Afternoon, professor and classmate. Today I'm going to present and explain about on an article called Mutational Analysis of BRCA1 and BRCA2 genes in Peruvian families with hereditary breast and ovarian cancer. First, I'm going to start with the introduction. Well, currently, breast cancer is one of the most common malignancies in women worldwide. Also, it is second to cervical cancer in Peru. So, in order to understand the article a little more, it is necessary to mention that the germline mutations in BRCA1 and BRCA2 are the two main genes predisposing to early development of breast and ovarian tumors. This mutation accounts for the majority of cases in families with high risk profiles, representing 26% to 84% lifetime risk for breast and ovarian cancer. It is also important for mention that the frequency of breast and ovarian cancer families with missense mutations, short deletions or duplications varies between 15% and 70% depending on the population studies. So about materials and methodology, first, blood samples were collected from 28 patients belonging to 18 families with a high risk for hereditary breast cancer. Second, these patients were identified at Oncosalud Auna Clinic, comprehensive cancer center in Peru. Well, they were referred for genetic analysis at the Centro de Genética y Biología Molecular of the Universidad de San Martín de Porres to gather information about some type of cancer and other cancer occurrence in the family and to take venous blood for ADN. Third, for DNA extraction, genomic DNA was extracted from peripheral blood leukocytes using standard methods. After that, DNA quality and quantity were assessed using a nanodrop spectrophotometer. For mutation analysis, Sanger sequencing of the entire coding region and exon intron boundaries of BRCA1 and BRCA2 was performing the later multiple primer sets were used for each exon to ensure PCR product amplification. Subsequently, sequencing results were compared with the BRCA1 and BRCA2 reference sequencing. Well, the MLPA technique was used to detect change in the number of copies of the BRCA1 and BRCA2 gene. In addition, in silico analysis was also carried out pathogenesis of novel missense mutations was predicted using in silico tools like SIF. Splice site alteration were evaluated using human splice finder. A statistical analysis was carried out. Well-described statistics were used for demographic and clinical data analysis, which was used chi-square of Fisher's exact tests were used to compare categorical variables p-value 0.05 was considered statistically significant. Results. For results based on the article identified 16 distinct mutations in BRCA1 and BRCA2 and include friendship, nonsense, missense, and splice site mutations. In our results, we mentioned that 4 families, three report that they have a mutation in the hairline BRCA1 and BRCA2, which we can see in our results table. In addition, a novel mutation was found in this study, which is located in exon 15 of BRCA1. This mutation was identified in family, and the woman was diagnosed with breast cancer at 36 years. Another result is about gene dosage alteration. Well, 2 genomic rearrangements were detected using MLPA in patients in family 2 and family 14. As seen in the image, the results are shown seeing a confirmation of both rearrangements was made that were analyzed in independent triplicate assays and subsequent. This was corroborated with a quantitative PCR using primers flanked in exon 7 of BRCA1 and primers the same for MP Lexon 10 as a normal control 2x. Endow's gene. Evaluation that both patients have a abnormal ratio compared with controls, reflecting the presence of extra copies. And the last in silico analysis results was shown that predict pathogenicity for novel missense mutations shows damaging effects and also identify alteration in spliced seeds were predicted to affect splicing. Discussions about identify mutation spectrum has been consistent with previously report mutations in other populations and further expand the mutational spectrum in Peruvian families. About prevalent mutations in Peruvian population, similar to other Latin American populations, highlighting the importance of region-specific genetic testing panels. Is so important the clinical implications since the identification of this mutation is crucial for genetic counseling and personally cancer risk management. Other point is about study limitations. Limited sample size, also lack of functional ACEs to validate the impact of novel mutations. Also lack of segregation analysis in family members due to limit ability of samples and potential presence of other genetic factors influencing the observer phenotype. Conclusions. Some conclusions about the article. 3 conclusions. This study provides insight into the mutation spectrum of BRCA1 and BRCA2 in the Peruvian population, identifying prevalent mutation is crucial for targeted genetic testing and appropriate cancer risk management. 3. Further research with larger cohorts is necessary to better understand the genetic landscape and improve clinical intervention. Thank you very much for your attention.